Good morning, dear students. Today's lecture is on diseases caused due to mineral selenium deficiency and other vitamin E deficiency. Selenium and vitamin E both are very good friends. They goes hand by hand. So for their functions also in body, we cannot separate them. In this slide, you will learn the diseases caused by selenium and vitamin deficiency in domestic animals. And we'll study details about the disease and their various aspects, such as etiopathogenesis, clinical findings, diagnosis, differential diagnosis, treatment, prevention, and control of selenium and vitamin E deficiency diseases. The selenium is a very essential nutrient for all animals. The diseases due to selenium deficiency are of worldwide distribution. It occurs in all countries and of very great economic importance to the farmer as well as to that country also. The biochemical role of selenium is it is a component of a very important enzyme, glutathione peroxidase. This enzyme, glutathione peroxidase, is a selenium containing antioxidant enzymes. Remember here, antioxidants are very much necessary to keep the cells healthy and live. Without antioxidants, there is death of the cell and death and cells will not remain healthy. This enzyme, glutathione peroxidase, is from erythrocytes of both cattle and sheep. Let us study the etiology of selenium deficiency. The deficiency occurs in many ways in the body. We will see each one by one. Selenium and vitamin E deficiency diseases of farm animals are caused by diets which are deficient in selenium and vitamin E. When animal is not getting the selenium in diet, definitely the animal suffers from disease. Another etiology, excessive polyunsaturated fatty acid called as PUFA in diet is also a conditioning factor. If there is more PUFA, less selenium and vitamin is available to the animal. Interaction between selenium and other trace minerals also occur. Selenium and vitamin E supplementation can provide protection against cobalt-induced cardiomyopathy. You know that cobalt deficiency causes pathology of the muscles of the heart that can be protected by selenium and vitamin E. Diets which are low in selenium because of selenium deficiency of soil on which they were grown. When there is deficiency of selenium in soil, the plant grown on such soil also deficient. When animal eat or graze such a forages, animal suffer from deficiency of selenium. That is universal for almost all minerals. Heavy neonatal mortality, unthriftiness or weakness in winter calves, goat and lambs, chronic diarrhea in calves, infertility due to fetal resorption in ewes, dietetic hepatosis, that is, necrosis of parenchymatous cells of liver observed in science, are some of the most important non-specific diseases which are often respond very nicely to dietary supplementation with selenium and vitamin E. The pH of the soil alkalinity encourages selenium absorption by plants. When plants are rich in selenium, animals also get sufficient selenium. The legume plants take up less selenium than grasses. So don't feed only legume. Mix the legume with grasses also so that animal get the selenium. Seasonal conditions, the weather conditions also influence the selenium content of the pasture and so also the selenium content of the animal also. We have seen many etiologies. Now let us see the pathogenesis of selenium deficiency. The dietary selenium, sulfur containing amino acids, and vitamin E act synergistically together to protect losses from oxidative damage. So, tissue oxidation is a must. Both vitamin E and the selenium deficiency in diet causes hyaline degeneration and calcification of muscle fibers. See here the importance of selenium and vitamin E for the health of the muscles. Regeneration of skeletal muscle is rapidly followed by invasion of phagocytes and regeneration of the muscle fibers. Vitamin E, chemically known as alpha tocopherols, is also serving as a biological antioxidant during normal cellular metabolism. Highly reactive form of oxygen, a free radical, are produced, and these include H2OT, hydroxyperoxidase, lipoperoxidase, superperoxides, various hydroxyl radicals, and a single oxygen. See how this oxygen free radical causes damage and how it is useful, that is governed by these chemicals. Selenium is very essential component of five selenoproteins: glutathione peroxidase enzyme, a deiodinase in liver and kidney. It is very useful in conversion of T4 thyroxine hormone to T3 and selenoproteins. Anemia, a very important disease, is also caused by this deficiency in swine, and it affects the bone marrow. And you know that synthesis of hemoglobin and RBC mostly occur in the house of bone marrow. Another very important disease is caused due to deficiency is nutritional muscular dystrophy. It is very commonly occurring in young cows, lambs, and foals. Vitamin A is active within the cell membrane as a lipid soluble antioxidant, scavenging free radical that get catch that otherwise might react with unsaturated fatty acids to form lipid hydroperoxidases. So such antioxidant phenomena occur. Polyunsaturated fatty acid PUFA contain high concentration of lanolenic acid. Increased concentration of PUFA in plasma increases the chance of free radical formation, and then free radical formation occur in cell. It is a cause of a tissue damage. Vitamin E or selenium deficient animals may be at a greatly increased risk of tissue oxidative damages when they are exposed to such diets. The myocardial and diaphragmatic form of the disease result in acute heart failure, respiratory distress, and there is a rapid death in spite of giving treatment because it affects a very important organ, heart. Acute muscular dystrophy results in liberation of myoglobin. This myoglobin is liberated into blood, and this myoglobin appear into the urea. The condition is called myoglobinuria, and the urine become a coffee color. Selenium and vitamin E deficiency in swine results in hepatic necrosis. The name of the disease is hepatic diabetica, degenerative myopathy of cardiac and skeletal muscles. We have already seen edema, microangiopathy in heart produce mulberry disease, and yellowish discoloration of the adipose tissue. The adipose tissue become yellowish. Now we'll study clinical signs and pathological changes due to selenium deficiency. It causes important disease, acute muscular anxiotic dystrophy, dystrophy of muscles, and affect an animal die without showing any symptoms because heart is affected. Another important disease named nutritional muscular dystrophy because deficiency of selenium, vitamin in nutrition, muscular, muscle, dystrophy, atrophy, degeneration, necrosis. Let us study this thing in detail. Cardiac form of disease. There is a sudden onset, all of a sudden, in the cardiac form. The lesions are present in the heart, diaphragm, and intercostal muscles. Important organs are affected. There is a depression, respiratory distress, dyspnea, nasal discharge, blood stained pulmonary edema, profound weakness. Animal is unable to get up, recumbency, rapid and often irregular heartbeat. The clinician on auscultation hear cardiac murmurs. There are extra sound from the heart, extra than lub and dub. The murmurs are observed. Rectal temperature, body temperature is normal, but sometimes may be elevated because of increased muscular work associated with more and more respiratory efforts. The caves are depressed with great discomfort of the respiration, dyspnea, tachycardia, increase in heart rate, and increased rectal temperature. Such symptoms are also observed in case of pneumonia. It is required to be differentiated from this cardiac form, and death of the animal occur within 24 hours, even if medical therapy is given, because heart is affected. We have seen the cardiac form, cardiac muscle is affected. Now the skeletal form. Here the skeletal muscles are affected. It is slower in onset than cardiac form, characterized by muscular weakness or stiffness of the muscle. Animal may be recumbent and unable to stand because the muscles are affected. Those that are able to rise on their own or with assistance show a muscle weakness. They again sit down. There is trembling of limb muscles and sometimes the stiffness of muscles. The supporting muscles of both fore and hind limbs become swollen. They become very hard and painful on palpation. There is great pain on palpation. The commonly affected muscles are gastrocnemius muscle, semitendinosus muscle, semimembranosus muscle, biceps femoris, and muscle of lumbar, gluteal, and neck region of the animal. If diaphragm and intercostal muscles are affected, there is great respiratory distress. There is great respiratory difficulty, dyspnea. Dysphagia is also observed due to involvement of tongue muscle. Cardiomyopathy occurs frequently. The heart rate may be increased, but the heart sounds are normal. Rate may be increased, but the heart sounds are normal. Animals with skeletal, nutritional, muscular dystrophy often respond to a rest and treatment within 3 to 5 days. When heart is affected, there is no treatment, animal will die. And here the animal can often stand and walk at their own. Such symptoms are also observed in many other diseases. Example, pneumonia, septicemia, and toxemia. It requires to be differentiated from this cardiac form and muscular form of the disease. In caves, dullness and severe respiratory distress accompanied by frothy or blood-stained nasal discharge is sometimes observed. Affected foal and calves are in lateral recumbency. They are unable to even sit sternum. They lie down laterally, unable to sit in sternal recumbency even when assisted. Again, they fall down. Heart rate increased to 150 to 200 per minute. Affected calves are thirsty and die within 6 to 12 hours after onset of signs. Temperature is normal. Subacute angiotic muscular dystrophy in calves, popularly known as white muscle disease. White muscle disease. The muscle loses luster, loses color, looks like with white streaks. And in young lambs, stiff lamb disease. The lamb walks stiffly. Affected animals will be in sternal recumbency, unable to stand, trembling of limbs, weakness, shoulder muscle and gluteal muscles are sore. These are the characteristic symptoms of the continuing the clinical findings. There is bilaterally major involvement of diaphragm and intercostal muscles occur in many cases and causes a great respiratory distress, dyspnea. And here the type of respiration is abdominal type of respiration. Heart rate increased. In severe cases, the upper border of the scapula protrude above the vertebral column and widely separated from chest and toes are spread. In foals, there is edema of the head and neck is very common. The horses may be presented initially with clinical signs of colic. Clinical pathology of the selenium and vitamin E deficiency. Characteristic here, the plasma creatinine phosphokinase CPK in muscular dystrophy of calves, lambs, and foals is of a very diagnostic value. Whenever there is muscle damage, the plasma creatinine phosphokinase level also increases. Always. It's a diagnostic value. Similarly, the SCOT activity is also an indication of muscle damage. Vitamin E and selenium deficiency produce a very important disease in swine, It is named as mulberry heart disease. The, the heart looks like a cherry red color, bright red color, just like a mulberry fruit. Deficiency of selenium and vitamin A occur in rapidly growing feeder pigs, piglets, and sows. The affected animal are commonly found dead without any premonitory signs because heart is affected. The stress of movement, inclement weather, or transportation will precipitate further acute death. Here, remember, the heart is affected and animal found dead with, without any premonitory signs. Other important disease in swine caused by selenium and vitamin E deficiency is hepatosis dietetica. Hepatosis मतलब कि necrosis of the cells of the liver. Exudative diathesis. There is lot of exudation from the blood vessels. Here also, most pigs found dead. Before death, the animal manifests symptoms like dyspnea, depression, vomiting, staggering of the gait, diarrhea, and collapse. There is great muscular dystrophy. There is esophagogastric ulceration. Ulcers are observed in stomach as well as esophagus. There is exudative diathesis. There is exudation of fluid from the blood vessels called exudative diathesis. The other animals also suffer from selenium and vitamin E deficiency. Some of the symptoms are observed in other animals are retained fetal membranes or retained placenta. There is blood abnormalities, anemia. In this, there is a decreased PCV and hemoglobin. Important organ, brain, myeloencephalopathy. It is also associated with vitamin E deficiency. It is exhibited by neurological defects, neurological symptoms, and degenerative myeloencephalopathy. There is generalized steatitis associated with vitamin E and selenium observed in horses and recently in fools. Steatitis means inflammation of the fat tissue. In this, there is fat necrosis called yellow fat disease, polymyositis, and muscular dystrophy. In this slide, we will see the necropsy finding or post-mortem changes in selenium and vitamin E deficiency. Here, there is bilaterally symmetrical myodegradation and is a consistent finding of this disease. Nutritional myocardial degeneration, dystrophy. The skeletal muscle degeneration is characterized by pale discoloration and dry appearance of affected muscles. We have seen that white muscle disease. So here in that the white streaks in muscle bundles are also observed and that represent a band of a coagulation necrosis, fibrosis and calcification. Peculiar finding of a white muscle disease. Cardiac muscle undergo changes similar to those of skeletal muscles. In calves, the left ventricle and septum are more frequently involved. There is a myocardial degeneration which extends through full thickness of the ventricular wall of the heart. It affects a very important organ, the heart. In this slide, we'll study diagnosis of selenium deficiency. We have seen the characteristic symptoms both in heart, liver, and muscles. By laboratory estimation of blood, there is increased creatinine phosphokinase enzyme and SGOT characteristic. Again, further confirmed by analysis of glutathione peroxidase enzyme, estimation of selenium content in soil, feed samples, and animal lesions. So this is important diagnostic estimation. Let us see the treatment aspect of selenium and vitamin E deficiency. The combined mixture of selenium and alpha tocopherol, that means vitamin E, is used in the treatment. The dose is given here in muscular dystrophy in cows, lambs, and foals. A mixture of selenium, that is sodium selenite, alpha tocopherol, is given. Depending on the species and type of production, the supplementation of selenium and vitamin E may be accommodated by several following ways. Add selenium to fertilizers for the soils. Selenium used as a top dressing of the plants. Just spray selenium content on the plants. Addition of selenium in the feed. Oral drenching of selenium products to the animal. Selenium pellets are also available that fed to the animal. Pellets are bit heavy, they settle down in the rumen and slowly release selenium over a long time. Intramuscular injections are also available, that is very useful and sure line of treatment. In the cardiac form, though treatment is given, chances of death are more because heart is affected. But in skeletal form, the treatment gives reward. Supplementation of selenium and vitamin E and injectable selenium products are also available. The dose is given here, various concentration is also given here for your information. Oral alpha tocopherol vitamin A products can also be given. Animal also require supportive therapy such as antibiotics to check the secondary bacterial infection either due to pneumonia or decubital lesion. Fluids and electrolytes also help in recovery. Properly prepared and stored hay grains and high quality of green forage should definitely ensure adequate supply of vitamin E. Let us summarize here what important things we have studied. Selenium is a very essential nutrient. It is a component of important enzyme glutathione peroxidase. It is a selenium containing antioxidant enzyme. Antioxidant diets which are deficient in selenium and vitamin E are fed, the animals suffer from deficiency. Or in certain geographical area, the soil is deficient in selenium. So naturally, the plants or forages or grass grow on such soil are also deficient. And when animal consume such grass, deficient grass, animals suffer from disease. Important disease we have seen mulberry heart disease in swine. The heart is of cherry red and just like a mulberry fruit. Other important disease in swine, hepatosis dietetica, that is necrosis of important disease we have seen hepatosis dietetica. It causes necrosis of parenchymal cell in swine. Exudative diathesis, edema is observed. Acute muscular angiotic dystrophy, generally the heart is affected. Sudden death in the animal. Even we have no time as a clinician to give treatment. Nutritional muscular dystrophy in young cows, lamb and foal. Subacute angiotic muscular dystrophy in calves called white muscle disease. And in lamb, stiff lamb disease. Laboratory diagnosis can be confirmed by increased level of CPK, creatine phosphokinase, and SUD, and is further confirmed by estimation of glutathione peroxidase. Combined mixture of selenium and tocopherol is a specific treatment of this deficiency disease. Dear students, you have observed this video. I have tried to simplify it greatly. It is also very much important. If you have any doubt when we'll meet here again, I will clear your all doubts. Thank you very much for observing this video.